All right. Thank you so much thank you so much for the introduction. Um, and thank you for the invitation from iSpark for inviting myself and Tzvetelina to present to you about CWD, chronic wasting disease. It's a pleasure to be here. It's an honor to um, be here and be worthy of your time to come learn about this important topic. So today, Svetlina and I are going to go over chronic wasting disease in British Columbia. What is that disease? Where do they come from? What does it mean for our deer population? And then we'll cover what the British Columbia Chronic Wasting Disease Surveillance Program is all about. Svetlina will go over food safety components of COPD. And then if we have time, we'll have fun with a little easy quiz at the end, just so that we can bring home the most important points of the presentation. So chronic wasting disease, what is it? It's an infectious prion disease. You might have never heard of a prion, but you might have heard of bacteria, virus, of parasites. Prion is just a different type of pathogens. So it's an infectious protein that causes disease in a number of animals. In the case of chronic wasting disease, it only affects deer species. So we're talking white-tailed deer, mule deer, reindeer, and possibly, most likely, caribou, moose, and elk. So it's a disease that causes neurological symptoms and it's progressive. So once the animal gets infected, they will become gradually sicker over time. And unfortunately, in all cases, the disease is fatal. There are no treatments or vaccine at this point in time available for this disease. So this might be the first prion disease you ever hear of, but I just want to give you a bit more context in the sense that many other diseases exist of that type that, are a source from an infectious prion. So you might have heard of Creutzfeldt-Jakob disease or mad cow disease in humans. That has a similar process for causing disease. So the prion is a misfolded protein that, when inserted into a body through ingestion or inhalation, will cause other normal proteins within the brain to become abnormal. And that process over time will create changes in tissues and cause disease. So here we have chronic wasting disease, but as you see, there's a all other list of prion diseases that exist. So chronic wasting disease is not a new disease. Well, in in it's new to us in terms of modern times. So it was first detected in 1967 in Colorado and has since spread to most other American states, several Canadian provinces, a few Northern European countries, and even South Korea. So on the left on this image here, you have what a normal brain looks like under the microscope. So you can see it's dense, it's all pink. All that pink is different types of cells. And that's what a healthy brain looks like. On the right side, that's a picture of what a CBD brain looks under the microscope. So you can see there's an obvious difference in color. There's all these sort of pale circles in there. And those circles are lesions caused by the prion. Another name for CBD is a spongiform encephalopathy. Very fancy word to say that it's a disease of the brain that makes it look like a sponge. So basically, the brain of the deer, will go from normal over time to a brain with very many small holes. And you can imagine that when you have holes in your brain, signs of disease will ensue. So chronic wasting disease, we talked about how well, it's in the name, it's a chronic disease and it's progressive over time. So animals will start off looking healthy, and over time, over months, over years, they'll develop signs of disease. A classic very, very sick animal from chronic wasting disease will look like this deer on the screen right now. So they'll be skinny, they'll be weak, their head will be low, they might drool a lot, they might be very thirsty, they might urinate more than normal. They might be stumbling, have a hard time moving. Um, but in every case, the last step of that disease process will be death, unfortunately. The tricky part is that most animals that are infected with the prion that causes CWD will look healthy. And that's, if there's one thing I want you to remember from today's presentation, it's this point. Most animals that are infected look healthy, or at least initially. So the picture in the slide prior was a picture from an animal that was in captivity. That animal was protected from cars, was protected from predation, was likely receiving some level of veterinary care, probably good nutrition. So that allowed that animal to live through the different phases of the disease development and look really, really sick. In the wild, if you're a deer and you are sick, most most likely you won't make it to the point where you would express that disease to that extent. So you'd get eaten by a predator, struck by a vehicle. Um, so most wild infected animal never look sick. The disease has a very long incubation period. So we're talking about anywhere from 2 to 4 years. And an incubation period is the period of time between the moment where you are infected to the moment where clinical signs are expressed. So when you first look sick or feel sick. So it can take years for an animal to express the disease. So for that reason, most cases are in deer that are anywhere from 3 to 7 years old. So they're infected, in their first year of life, let's say, and then 2, 3, 4 years pass. And by the time they're 3 to 7, then they'll look sick. But it doesn't mean that before they look sick, they weren't infected. So because it's such a tricky silent disease, the only way to know for sure whether an animal is infected or not is through testing. And we'll talk about that at length in the next little bit. So here's a little quiz. You're welcome to put an answer in the comment section or put up your fingers to the screen with the number of which you think is the answer. My question is, which one of these 4 deer is more likely to be infected with chronic wasting disease? I see Adam and Jerry on the screen. I don't mean to put you on the spot, but do you have an answer? I can't see comments. I'm just realizing that.
We have a lot of two.
Okay. Yes. So a drooling deer that looks a bit spooked. The first one is skinny, the second one's drooling, the third one was unfortunately hit by a vehicle, and the fourth one is especially healthy looking. So you're right in thinking that the two might be sick, because that amount of drooling in an animal is not normal. However, the question was which one is more likely to be infected with CWD? And that was a trick question because the answer is that they all are as equally likely to be infected with the disease because, as we just talked, the disease is silent for a long time. So you could have number 4, a very healthy buck that's infected and won't declare evidence of disease until, a year or two from now. So what I want you to remember from this is that it's not because you see a healthy animal in the wild that you can know for sure that it doesn't have CWD. We have a lot of people that come to us and say, "Oh, yeah, I harvested a deer last week. I'm thrilled. It looked healthy. It doesn't have CWD. I don't want to test my head." That's a flawed logic because the disease is silent and you cannot know until you test. And then the opposite is true too. People will see very, very sick deer. There'll be a skinny deer that's stumbling around, and they'll go, oh, must have CWD. And in reality, once we tested, it doesn't, but it had no teeth left, and it was probably 15 years old. So prions are very interesting pathogens. Compared to viruses or bacteria or parasites, prions are practically indestructible in the environment. And that's what makes this disease so difficult to manage once it's introduced in the environment. What I mean by indestructible, you might remember in the COVID times when um, we would leave our packages outside the door, or we would use hand sanitizer and it would deactivate the virus, and then we'd be safe to handle that object. That doesn't work for prions. They are so resistant to cooking, radiation, freezing, and most chemical agent that they're practically indestructible. So because they cannot be destroyed, it also means that they can accumulate in the environment over time. And we're talking about the capacity of prion to remain infectious in the environment for 10 years and beyond. Studies aren't old enough yet for us to really know how long a prion can persist in the environment and represent a risk for infection for animals. And these studies have been done on a variety of materials, plants, plastics, cement, dirt, hay, and they've shown that the prion lives in those different surfaces for a very long time. That's very different from bacteria and viruses and parasites, where, if it's a bit too hot, the bacteria will die, or it's a bit too cold, the virus will die, or it's too dry, the parasite will die. Prions are practically indestructible. So how is CWD transmitted? Well, it can be transmitted directly between animals through fighting, touching from animal to animal, socialization, even maternal transfer through close contact between different animals. But then the tricky thing is that it can also be transmitted through contact with contaminated environment. So we're talking about a deer that's healthy, comes around the corner, munches on a blade of grass that has a prion on it, boom, infected. So that's what's scary about the disease. It's it because it is indestructible in the environment, it remains infectious and it represents a source of infection for animals that come along. So we were just talking about disease transmission. There are several risk factors for disease transmissions that I think it's very important to know what those are, because that gives us an opportunity to act upon those and prevent the spread of CWD in the environment. So risk factors for disease transmissions are anything that attract a high number of deer to one specific area. Because if you have a lot of animals that concentrate in one area, they'll salivate, they'll urinate, they'll defecate, and through all of these actions, they'll shed prions. Given that they're infected, they'll shed prion into the environment. And sort of, if there's a mineral lick there and they're shedding prions, they can basically create a hot spot of infectious um, potential in that small area. And then animals will come afterwards visit and then can get infected there. So anything that artificially increases the concentration of deer in one spot can be considered a risk factor for disease transmission. So we're thinking about mineral licks, urban deer situations where there's an abnormally high number of animals in town, and then any sort of attractant, either um, conscious or not, such as, some people will bait deer for hunting, others will have hay out for the horses, but the field's not fenced. So it can be a number of things. So now let's look at risk factors when it comes to disease spread. So we know what um, risk factors have to do with transmitting the disease from one animal to another. Now let's look at what risk factors have to do with how the disease spreads across the landscape. So first one is through the transport of carcasses that are infected or byproducts of animals that are infected. So we're thinking of scent products used for hunting that are based on um, the actual urine of animals, like a natural product directly from animals. There's always a potential that those products could contain prions. And then same thing with animals that are being transported after harvest. Um, you won't know until you get your head tested that the animal is positive for CWD. So moving carcasses across the landscape comes with the risk of moving the disease across the landscape and introducing it to novel areas where the disease hadn't been present or detected before. A second risk factor for disease spread is through hay transport. That's a tricky one because we know there's been abundant studies on this. We know that if there are prions, so it's a little protein in the ground where hay grows, the prions will get into the roots of the plant and then come up the blades as the plant grows. And then picture the scenario. The hay is harvested, it's put up on a transport truck and then shipped across the country or across province lines or across regions. That hay that contains the prion is a source of infection. So it could be fed out to animals in a new area. Animals come, have a good snack, they're happy, but then they become infected. There's currently no way of knowing whether or not that hay contains prion. So what's recommended is to avoid sourcing hay from regions that are known for being CWD positive. And then the third risk factors for disease spread, something that we don't really have control over, but and is somewhat negligible, I think, but still interesting. So I wanted to mention it, is through scavengers. So a deer dies, coyote comes over, has a meal. That deer is infected with CWD. The meat is ingested, the prion is ingested, it lives in the intestines of the animal. The coyote moves on with its day, goes somewhere else, defecates. Um, Live prion, infectious prion particles will remain in those feces. Um, so it's one other way that the prion can travel across the landscape. There's new research that shows that the vast majority of those prion is deactivated through the digestion process, but the fact remains that there still is a possibility to transmit infectious prion over the landscape through scavengers. So we're thinking of scavenging carnivores, birds, but also ticks. A tick can have a blood meal from, isn't that lovely? Take a blood meal on a deer that's infected. The blood itself contains prion. The tick falls to the ground, and there you go. The prion is moved across the landscape. So, just to illustrate how COPD has spread over the landscape over the past few decades, I have here a series of slides. So you can see here Colorado. That's where the first cases in 1967 were detected. We don't know where the disease came from. It popped up in a research facility, in a government research facility in the 60s. There's many hypotheses as to where the disease came from. There's the one of the sort of spontaneous mutation. A glitch happened in that animal's DNA, and then boom, it coded for a protein that was abnormal, and that caused disease. Another hypothesis is that that research facility, I think decades before housing deer on their pastures, had housed sheep with scrapie. And scrapie is a prion disease of sheep. So there's thought that perhaps there was sort of a, crossed wires that made the protein for sheep, jump species barriers, and then adapt for deer. And then boom, we had a new disease. But we don't know. And at this point, I don't think we'll ever know. But I guess it's the big question. Where do they come from and how do we prevent it from popping up somewhere else? It exists too. CWD exists in Europe, and they've shown that it's a different strain that they think has spontaneously appeared um, in moose and reindeer. So we don't know yet. Hopefully one day we know so we can prevent it from happening again, but that's just what happened. So each slide that I'm going to show you here is a year, and we're going to do a progression through time. So you can see the gray is CWD in free-ranging population, and then yellow is in farms. So this is prior to 2000. In 2002, we had the first case pop up in Saskatchewan. So this is 2004. Now we move forward in time. You'll just see it spread slowly but surely. It popped up then in Alberta in the late 2000s. And then over time, you can see more and more cases were detected in more and more new areas of North America. And once it's established in an area, as far as we know, there's no way of eradicating it. You can only hope to slow the spread of the disease and manage it that way. So moving on to 2022, 2023, and then sadly in 2024, we had our first detection of CWD in British Columbia, in southeastern BC and Cranbrook. So it was sort of, I don't think we could have avoided it just because we have deer crossing, migrating all over. And for a long time, we were protected by the Rocky Mountains, but we think it came up. We don't know where it came from, but there's a likelihood that it came either from across the Rockies, from deer migrating across, or from Montana or Idaho. And this is the most recent map available of the distribution of CWD across North America. So we'll take a break from disease spread and we'll talk about human exposure risk factors. So how can you as a person be exposed to CWD in life? So the first one is through exposure contact to meat, game meat, and viscera, either handling or consumption. Second one is through butchering animals that are infected with CWD. Obviously, at that point in time, you wouldn't know. And then the third one that's less common, but relevant for some part of the population, is through laboratory work, through handling of deer tissues. So now that you know that eating and handling game meat is a risk for CWD infection, I'm sure you wonder, is CWD infected meat safe for human consumption? So I'm not the expert on this. This is a topic that we like to reserve for human health authorities. Tzvetelina will have a bit more to share with you about this specific topic. What I can tell you, though, is that there has been no human cases of CWD reported yet. So we know other prion disease do occur in people, but CWD itself has yet to be recognized as a disease that can infect humans. But the research is a bit inconsistent. So at this point in time, all we can say is that health authorities recommend that known positive animals not be consumed. The other thing that's very important in terms of consuming game meat is that cooking does not inactivate the prion. So you can cook it, you can char it, you can make sure you freeze it before you cook it, you can bake it. None of that will inactivate the CWD prion. So if it is in the meat, There's no way of removing it before eating it. So positive animals should not be eaten, and cooking does not inactivate CWD prions. And this goes, that picture had meat on it, but this goes for most tissues in the animal. So this is a graph of the concentration of prion across different tissues. So the highest risk material, the tissues that will have the highest concentration of prion within an animal that's infected, will be brain, spinal cord, and other nerves. And then the GI tract, reproductive organ, and the meat itself will have a bit less prions. And then blood, saliva, feces, the velvet on the antlers, and the urine will have less. But keep in mind that it's a gradient, but it still means that all these tissues have prions. So if you wonder, I understand meat can have prion in it, or brain can have prion in it, but maybe liver or kidneys. No, they all, if an animal is infected with CWD, we can assume that all tissues can contain some concentration of prions. So we've talked about the risk factors. Let's talk about the impacts. What does it mean to have CWD amongst us? Well, there's direct impacts. Those are on animal health, obviously. It causes disease in deer that affects their health, it affects their overall welfare, it affects their population um, health as well. Population have been known to decline once CBD is introduced in the population. And then there's always a potential for human health risks that I've just discussed. Indirect impacts are equally important. We're talking about food security. CBD could impact our food security in the fact that, one, there might be less deer on the landscape one day, and two, the meat that we harvest might not be safe for human consumption. This has the potential of influencing traditional practices, cultural heritage. It affects how hunting is regulated. It affects ecosystem on more levels than just deer, right? You have less deer, there's less of a prey base for predators, and then it's a snowball effects on an ecosystem level. Also has economic impacts and psychological impacts. So now that we've gone over what CBD disease is, let's talk about what BC is doing about it. I just want to make clear that this presentation is angled towards the surveillance program and not disease management actions. If you're interested in learning more about management actions, there's plenty of resources online, and you can reach out to the government of British Columbia directly for those questions. So here we're going to talk about the surveillance program. So I had the privilege of being involved with this for a few years now. Surveillance is the attempt of detecting the disease before it becomes widespread. So we're trying to look at animals, we're trying to find the disease, we're trying to figure out how much of it there is, how serious it is, where it is, so that we can then make decisions on how to organize a response. So the sooner we have information and the better that information is, the better decisions we can do in terms of planning a response. Surveillance, the goal of it is to support hunters, to support community. It helps maintain safe, sustainable access to wild game, and it protects the economic and cultural value of hunting tradition. Surveillance is also important because, like I said before, the COPD has consequences on people, but it also obviously has consequences on animals. So the earlier we can find out where it is, the earlier we can do something about it, and the more we can protect our wildlife. So the BC CBD surveillance program started a long time ago. They started in 2002 or just around there, and have over time sort of amped up that program to what it is today. It is a free service that allows you to get your deer head tested for CWD. It's voluntary in most areas, but in high risk area in region 4, and I'll show you a map, it became mandatory once CBD was detected here. So the surveillance program is focused on, so far has been focused on high-risk region and along the provincial borders because we were trying to detect when it would jump from Alberta or Idaho or Montana. Um, However, it doesn't mean, now that you know that CBD can be transmitted from region to another region through different means, through hay, through movement of carcasses, birds, it doesn't mean that CBD won't pop up in the middle of the province. So the regions that are vulnerable right now are the ones that have low sample size. I'll show you stats where the program hasn't really isn't strong yet. So that means that disease in those regions, we have people from different areas of BC here today. It could mean that the disease is already in your region, but we wouldn't know about it because the testing hasn't been done. So the main goal of the surveillance program is to detect the disease early so that we can plan a response. The second goal is to support communities with outreach and education, just like we're doing today. Bring awareness about the disease so that we can all work towards the common goal of slowing the spread of the disease. Okay, so what does it mean to have a surveillance program in British Columbia? So let's just walk through the different steps of the program. What needs to happen between the time where you submit a head, you harvest a deer, and you have a head, to the time where you can go online and look at your result and see whether or not the head that you submitted was positive, negative for CWD. So first you harvest a head. Oops. Harvest a head. Second, very important, you fill out a card that is oh so important because that's how we'll have your information so that government can contact you in case you get a positive result on your head. Um, and it's yeah, it's important to be as thorough as you can be on that card when you fill it out. So once you have a head with a card on it, whoops, I keep clicking the wrong side. You can deposit it in one of the many freezers that are across the province. They're specifically for this purpose. We call them drop-off freezers. Um, if you go online, just on the Government of BC website, you can find a map where all the CB freezers are. And if there isn't one in your region and you'd like there to be one in your region, you're more than welcome to contact the COVD program and have a discussion about it. Once the heads are in the freezer, we have contractors that will go around the region and pick up the heads and bring them to a warehouse where we have technicians that will sample the heads, extract the samples that are needed for lab testing. So we're talking about lymph nodes, tonsils, and in moose and caribou, the obex, so a portion of the brain stem. Once those are extracted, labeled, organized, all that. They're shipped once more to the lab in Abbotsford, the animal health lab, the provincial animal health lab, where those samples are actually tested. And then at that point, those results are uploaded to the website where you can now go and check your results. The way you find your result is with the number that's on the card here. So in this example is 1250. Make sure that you make a note of that number or keep that portion of the card with you so that you know what to look up when you go on the website to look at your result. So given that this is a multi-step process, there will be a lag time between the day that you drop it in the freezer and the day that your result will be available online. And we're doing the best we can, but you should expect a delay of anywhere from 2 to 3 weeks. So I was talking about certain tissues that are extracted by technicians for submission to the lab. Those will vary based on the species. This is a lot of information. If you're not keen to extract samples yourself, you can just submit a whole head. There's instructions. submit the whole head in the bag, move on with your day. If you're interested in learning how to sample these different tissues, there's plenty of resources online, and sometimes the BC CDB program will put up sort of wet labs to teach people how to extract those tissues themselves. So for mule deer and white-tailed deer, what we're looking for are certain lymph nodes, tonsils, and so you can either submit the whole head, those samples themselves, or just a lower jaw if you want to keep the skull with the antlers. The lower jaw contains everything we need as long as you leave the soft tissue attached with the jaw itself. If you harvest an elk, a moose, or a caribou, that's a bit different. It's the lymph nodes, but also the base of the brain. So a section of the brain stem. So in that case, we ask that people submit a whole head. Like I said, if you're interested in learning more about how to submit samples, how to extract them yourselves, there are plenty of resources online. These are two videos that were produced by the BC CBD program itself. So let's travel in time and go back to 2024 when the first cases of CWD were detected in the province. So this happened in January 2024. We had two cases, one in Cranbrook and one a bit south. So that was the first time CWD was detected in the province ever. This triggered a immediate response by government. They established what we call the CBD management zone. So on this map here, you can see this is region 4. We have Cranbrook, Invermere, Revelstoke, and then within region 4, there's this area here that is red, and this is what we call the management zone for CBD. This came with a set of new rules to try and decrease the likelihood of CBD spreading beyond those boundaries. So there are new carcass transport regulations that apply to this area. Basically, the brain and the spinal cord, excluding the tail, must not leave the CBD management zone. So you can bring it to a CBD freezer within the region, but you cannot bring it to a CBD freezer or anywhere else beyond that region. And then it came with recommendations for best practices around carcass disposal. So the ministry recommend that animals be deboned at the kill site and that all the parts that you don't want to keep with you be left on the site. Or brought to a landfill or a designated disposal site within the management region. And then the third thing this management zone comes with is a mandatory order for CBD testing. So for that region, for these management unit within region 4, mandatory CBD testing became a condition of the hunting license. That calls for heads to be submitted within a week of harvest. So after this, let's go back to our map. These are our first two cases that were detected in early 2025. This is last year. We had 4 more cases that were all detected around Cranbrook. And then this year, we had 3 additional cases around Cranbrook and then within a new area that popped up. So CWD, the BC CWD surveillance program, like I said, started in 2022, but really ramped up its intensity in 2023 when CWD was basically knocking at the door just south of us in the States. So let's look at how the program has been doing since 2023 until now. So we've received a total of 10,000 heads that have been sampled for testing. 83% of those, so a vast majority, come from the mandatory zone. So that section of region 4 where cases have been detected. and about 17% came from elsewhere. So elsewhere in the Kootenays, but also beyond in the rest of the province. In terms of species, most are white-tailed deer, and we get quite a few elk and quite a few mule deer and a small proportion of moose. So of those 10,000 heads, we've had 9 positive cases so far, and that represents about 0.1% positive. So you can see all the green here is negative cases, and that little sliver of red is what represents the positive cases. So the prevalence is still very, very low as far as we know. But the more we sample, the more information we gain, and the more we can paint an accurate picture of the situation. So this is a representation of head submissions per region in British Columbia. So the Kootenays, which again includes the management zone where CBD testing is mandatory, represents 88% of the total head submission for those 3 years. All of the other ones, all of the rest of BC fits into this tiny little pie section. So here we have numbers. For example, the Cariboo region, over 3 years, we've only tested 77 heads. In Ominica, 25 heads. Skeena, 15. So you can see now how, even though the sampling and surveillance effort is very strong in the management unit in the Kootenays, we could be missing it in other sections of British Columbia. And that's why it's important to test heads, even if you're not in a section of the province where CBD is known to happen, because that's how we find it, and that's how we stay safe, and that's how we protect our wildlife. So I realize this is a bit of a gloomy presentation. It's a lot of information, most of it is scary. But what I want you to know is that we can do something about it. And if we all do a little bit, we can make a major difference on how CBD behaves in British Columbia in the future. So what can you do to help? The first thing is that we need to keep hunting. Hunters are the most powerful tool that we currently have in managing CBD on the landscape. It helps remove positive infected animals from the landscape, and it also is our main source of information in terms of disease surveillance. One common misconception, or one common, I guess, misguided reaction is to go, oh, there's CBD in the deer population where I want to hunt, so I'm not going to hunt there anymore because they're sick. That's the last thing we need to do, because if we stop hunting populations that have CBD in them, the disease will spread, it will become entrenched in the landscape, and then there will be no going back. At this point in time in British Columbia, we're in a very, very special situation where we're early in the disease spread. Now is the time to do everything we can to prevent it from spreading. So the risk of harvesting a positive animal at this point in time is extremely low, sorry. Even in the management zone, you saw we sampled 10,000 heads and we got 9 positive cases over 3 years. The second thing you can do to help slow down the spread of CBD is to report sick deer. That is such important information. If you see an animal that looks sick, skinny, behaving abnormally, there's no way of knowing if that animal has CBD or not. But you can report it to your local conservation office service, sorry, or your local biologist, or call the BC CBD program and report those animals so that there can be follow-up on them. The other thing you can do is follow carcass disposal best practices that I just mentioned. Those ones are strongly recommended for the management unit, but you can apply those practices everywhere, anytime you handle a carcass, just in case that carcass is positive. Because it's better to be um, proactive than not. And then avoid feeding or baiting wildlife to not create those artificially high concentration of animals. Follow carcass transport rules, just like I mentioned for the mandatory zone. And then Tzvetelina will tell you more about this. Follow meat handling safety guidelines to protect yourself and your family. Avoid sourcing hay or feed from CWD endemic regions. We can think of hay from Alberta. Stay informed and share accurate information. Come to webinars like you're doing today. Use synthetic scent products. Avoid the ones that use actual urine from animals. Use those that are synthetic that cannot contain CWD. And then last but not least, sorry, participate in CWD surveillance. And there's many ways of doing that. It can be through submitting the head that you harvest, and it can be too through sharing information, but also if you have an interest in CVD surveillance, and if in your region there's not already a setup, there's not a system, there's not a freezer, there is no outreach in your specific region, please reach out to the BC Wildlife Health Program. Let them know that you're interested in supporting a surveillance for CWD in your region, and then they'll work with you to make that happen. There you go. That's it for my section. So if we have time after Tzvetelina's section around um, meat handling guidelines, then we can come back and do a little quiz.
Thank you so much, Emily. That was
You're
great.
welcome.
Okay, can everybody see my slide?
Yes.
Okay, yeah, thank you so much, Emily. So I will be presenting on the food safety aspect of the chronic wasting disease aspect. So my name is Tzvetelina Petkova, and I'm an public health officer with First Nations Health Authority. So I would like to start with the land acknowledgement. I'm grateful to work, live, and play in the traditional, ancestral, and unceded territory of the Ktunaxa, the Secwepemc, and the Sinaiax people. So just looking through the agenda, I will be doing a brief introduction of our environmental public health service program, going over the human health implications and precautions, testing in CWD management zones, safe handling in CWD, safe handling of animal tissues, cross-contamination prevention, equipment cleaning and then disinfection, and then going over a couple of gaps and inconsistencies that I have found. So I am an environmental health officer in the Environmental Public Health Service with FNHA, and we cover 9 core programs, as you could see in this graph. And today I will be focusing on the food safety area. So to date, there is no direct evidence to suggest that CWD may spread to humans. However, as a precaution, it is recommended that people do not consume any part of animals that have tested positive for CWD or from untested cervids where CWD has been found. So there's no human cases. Lab models where human brain cells and petri dishes did not appear to become infected after direct exposure to CWD prions. However, if you're giving hunted cervid meat, it's always recommended to ask if the animal is hunted in an area where CWD has been found and if it's been tested. So although there is no documented transmission of CWD to humans, that shouldn't mean we should assume that will stay that way. And since the existence of a potential risk cannot be definitively excluded, I'll be going over some key recommendations found in various jurisdictions that are dealing with CWD and research papers to reduce exposure to CWD prions. So the first key recommendation is to identify sick animals and seek testing. So as in Amelie's presentation, testing is the only way to confirm CWD in cervix because animals are asymptomatic even after infected. Symptoms in clinical CWD cases can be broad and can be seen in other diseases, and there's no telltale signs of CWD infection in carcass after butchering. So in the field, to be safe as possible and decrease potential risk of CWD exposure, comes with safe handling of high-risk tissues, especially if the CWD status is unknown. So the following actions can be taken, and this could be applied for other diseases in game meat. So prior to the removal of the high-risk tissues, which is brain, spinal cord, lymph tissues, hunters should don personal protective equipment. So this would include eye protection, disposable gloves, and disposable gowns. Have dedicated equipment. Example, bone saw should be used for removal of high-risk tissues, and do not use that bone saw or equipment to process edible tissue. Cool the carcasses rapidly after kill, and that could include removing as much hide and skin to facilitate cooling. When you're removing internal organs, take care not to puncture intestines or stomach. And after you're done handling with the animal tissue, wash hands thoroughly. So that would include using potable water and hand soap. Don't use hand sanitizers because they're not very effective, especially when you have organic material such as meat, blood, or fat, and they're not proven to be effective against deactivating prions. So cross-contamination, that could occur between infected and healthy cervix or between cervix and other animals during processing of the meat via the cutting tools, surfaces, or other equipment and storage. So during processing, it's very important to keep the meat from each carcass separate, protected, and labeled. So game and non-game meat ideally should be kept separate to reduce opportunities for cross-contamination during the processing. And that's especially important for carcasses of unknown CWD status. Batch or multi-carcass processing of meat with unknown CWD status is not recommended, as potentially infected meat can contaminate the meat. It's also recommended that you have single carcass processing, and that's because it's easier to track animal to animal, and it ensures that each hunter gets his or her own meat. And that greatly reduces the risk of cross-contamination. And ideally, only tested animals are processed. The reason why we want to avoid cross-contamination and prevent it is because prions are not destroyed by cooking temperature. So you don't want to infect non-contaminated meat with prions. So when you're done with your cutting tool, surfaces, or other equipment, the first step is cleaning. So the most common recommendation is to scrub with normal dishwashing detergent, and that's to remove visible tissue, blood, fat, or other organic matter. Just as a general sanitation where bacteria and viruses can hide in organic matter, so can infectious CWD prions. And scrubbing is important because it can reduce 99.9% of microbes or 3-log reduction, and therefore optimizes sanitation and disinfection, which follows. Another recommendation is to keep meat processing equipment moist, and that would prevent residue from drying and sticking to surfaces because otherwise it would be harder to remove the organic matter. So after cleaning is disinfection. Different contact surfaces and materials do require different contact times for prion inactivation. For example, a complex meat grinder would require a longer contact time than a knife. And this has to do with interaction with porosity, how surface material interacts with disinfectant, and it also depends on how good the cleaning step was at removing or matter. The most common current recommendation is using 2 to 2.5% sodium hypochlorite bleach solution with a contact time of 60 minutes. And that's in particular important for porous and hard to clean equipment. After the 60 minutes contact time with the sodium hypochlorite or any disinfectant, make sure that you're rinsing it with potable water, especially since that bleach residue and bleach of that concentration is a very strong oxidizer and can damage equipment surfaces over time. The strength for this bleach solution is also not considered food safe and can then potentially leave residue on the food. So how would you make a 25,000 parts per million or 2.5 sodium hypochlorite solution for disinfection? So you would make it using an unscented household bleach, which is commonly 5.25 sodium hypochlorite. Mix 2 cups of clean water with 2 cups of bleach, and that would make 1-to-1 ratio with a concentration of 25,000 parts per million. Just make sure that you're making new solution every time you require disinfection because bleach solutions lose strength quickly. And there's also other factors that degrade the power of bleach, which can include organic load, contamination, temperature, direct sunlight. When you're making your bleach solution, make sure you're wearing personal protective equipment because bleach irritates the mucous membranes and respiratory system and can react with equipment. So make sure you're wearing your gloves, eye protection, working in a well-ventilated area, as well as something to protect your clothes. There are other alternative disinfectants that have been proven effective as well, but they also have some limitations. Highly alkaline solutions such as sodium hydroxide or potassium hydroxide, along with heat treatment, are known to be effective against prions, but they're not generally recommended for safety issues. Phenolic products such as Wexide 128 can provide a CWD prion inactivation, for example, for steel wires, but it is quite pricey and sometimes hard to find. A 5-minute soak with Briotech is shown to remove infectious prions from knives, but not from cutting boards. Virkon S, which is a potassium peroxymonosulfate, removes infectious prions from knives and cutting boards, but not shown to be effective for meat grinders. So you could see these variations is why it's recommended to use the 25,000 parts per million bleach solution in 60 minutes, because right now it's better be safe than sorry approach, and it removes some of the guesswork until we have a better idea. So there are some gaps and inconsistencies, and these are some of the ones that I found. So as Emily mentioned, CWD has been around for decades and prion diseases for even longer. And it could take years or even decades for symptoms to show up in cases of infection. And even though there appears to be interspecies barrier to human infection, there's still a lot of variables that can influence prion diseases. There's no one set in stone cleaning and disinfection recommendations. Various jurisdictions have different bleach concentrations and contact times. Alternative disinfectants, is there any ones that are better effective than others? Maybe not as harsh. So there's still a lot of questions around that. And food back donations. So right now, there's just advisories against using untested donated meat. So just in summary, as CWD spreads, there's going to be increased human exposure. However, right now, the greatest risk to human health from current data is food security. And that's because humans and indigenous communities rely on harvesting cervid as of a source of protein. So looking in the future, improved understanding of human health risk, herd protection, cleaning, and disinfection will help straighten how we face CWD over time. So here's my last slide, and this is just contact for our environmental health team and a website of what the environmental public health team does. Thank you very much, and here's some more references for further reading.